We will start in about a minute to give everyone a chance to join the webinar. Hello, everyone again. Thank you for taking the time to join us today for this exciting webinar being brought to you by Syntica. My name is Ivy, and I will be your host for today's webinar. Our webinar today is entitled M-finite applications of the M-series MRI. Today's webinar will be given by Dr. Kara Wendell. Kara is a product manager at Syntica Instrumentation. She specializes in Aspect M-series compact MRI. Kara received her PhD from UC Irvine, where she studied how early life experiences shape brain circuits. During her PhD, she utilized MRI and viral tracing to map circuits within animal models. She followed her passion for science and cutting-edge technology by joining Syntica Instrumentation, where she now works closely with researchers to implement and optimize the use of MRI in laboratories. Before we begin, I'd like to briefly mention a few housekeeping items. We anticipate the presentation will be about 40 minutes, which should leave us time for some questions. Please submit your questions in the Q&A chat box. Throughout the presentation, we will go through them at the end. We will also create a transcript of the questions we do answer during the session, as well as those that we may not have a chance to address. This transcript will be made available afterwards. I would also like to mention that how to sign up to use the M5 at the UIC will be discussed at the end of the webinar. So please note that we are recording today's session. So if for some reason you lose connection or can't hear anything, you will have access to the video file to review later. Without further ado, I am pleased to turn this over to Dr. Wendell for today's webinar.
Thank you, Ivy, for that generous introduction. And thank you all for attending today to talk about the infinite applications of the M-series compact MRI. So just a quick outline, we'll review the M-series magnet design and the user experience. We'll also then go to a poll question, and the main focus of today's webinar will be the application areas, which range from anatomy, morphology, cardiovascular biology, cancer. So we'll review all of that shortly. So a little bit about the M-series. So essentially the M-series is a permanent magnet. So there's no special infrastructure or cooling that's required to house this magnet. And so this is partially related to the fact that it's compact and self-shielded. So you may notice that these magnets are placed in existing imaging facilities within vivariums. They're pretty much able to fit anywhere. And within that space, there's no interaction with other metal components. There's no interference from other imaging systems within the same room. Even within the external fringe field, which is shown on the red as the red line on the floor, you're able to have metal components like vaporizers, other equipment, surgical tools. So you don't necessarily have to worry about the external fringe field, any interaction with metal components. And so oftentimes, like at the UIC at University of Minnesota, you'll see it's placed within an imaging facility. Alternatively, there's times we place them within the animal facility. So there's no, again, interaction with any other equipment in the room. You're able to use your cell phone, laptops, any other sort of electronic equipment is okay. And you can also operate it very quietly. So you don't necessarily have to worry about any stress, either if you place it within your lab or within animals in the room. Even with the operation overnight, you can turn it off so it doesn't have any noise that affects animals that are housed in the vicinity. And systems are typically installed within about a day. They're loaded off the loading dock, placed onto a set of wheels, and then wheeled into place. So installation occurs very quickly. Training occurs in about 2 days. And then once the UIC managers should be able to train you fairly quickly as well. So the components of the system are quite minimal and very compact. So we'll review the M5, which essentially has the electronics cabinet and the user workstation or the computer, which is shown here. You have the magnet itself, which is shown in the front, and then you have animal handling systems. So in this photo, the animal handling system is placed inside the magnet, but you have different animal handling systems for both mice and rats. And you'll see when we discuss the animal handling systems that we do split by different species. So our animal handling systems are also fully integrated both with a bed and a coil. And so we typically delineate between mouse and rats. Oftentimes our rat beds can be used for slightly larger or smaller species of the same size like hamsters. And we also provide anatomically specific coils. So we do split into head and body coils. So if you do use the system at UIC or another M-series system, you'll see that we oftentimes split into a mouse head coil or a rat body coil. We also have larger body coils for animals of a larger size. Each of the animal handling systems comes with heating. So this is one way that we just maintain animal welfare. Also, physiological monitoring is integrated into the system within the beds. So we do monitor ECG, heart rate, respiration rate, as well as temperature, just to maintain your animal welfare. So this is always important when applying for IACUC or animal approval protocols, just to maintain that the animal is doing well inside the magnet, and any issues you can see from the outside, and then remove the animal if needed. We also have anesthesia delivery directly in through the magnet and active gas scavenging, so you're not inhaling any excess isoflurane or sevoflurane that's being delivered to the animal. And the system really is designed to be extremely user-friendly. So on the front flange, you may notice there are different ports to connect the animal handling systems. That includes the heating, the anesthesia, as well as the monitoring ports. Each of these ports are individual and different. So if it's not the correct port or if it doesn't fit, you can just go down the line. So you don't need to necessarily label the cords or anything like that, just to give you a slightly more streamlined user experience and add to that ease of use. So since the magnet is a permanent magnet, it doesn't need electricity to maintain the field, so you can just turn it on when you're ready to image and anesthetize your animal. Once the animal has been anesthetized, you can place them inside the animal handling bed and attach them to the bite bar. So from the bite bar is where anesthesia is delivered. And as the animal's pulled into that nose cone, we also undergo active gas scavenging from that point. So you're not inhaling any excess isoflurane. The tail is also pulled through the small hole in the back. This is just an example of ways that you can do dynamic imaging if you want to inject agents while the animal's inside the magnet. So the coil has 2 electrical connections, one of which is this copper wire, and the second one does all the tuning and matching for the body coils. And so it's really adds to that streamlined user experience. Additionally, as the animal is loaded into the bore, you may notice in this example of the M3 that you have markings along the bore. In the M5, you'll see that there is markings along the animal handling system. This allows you to have an understanding of the positioning of the animal inside the magnet. So if you're imaging something like the heart versus lower down in the hind limb, you would use slightly different positioning as just a way to understand where you are in the magnet, so you don't have to make quite as many adjustments going forward. After the animal's been placed into the magnet, you're pretty much able to transition to working with the software. So the software interface and the user experience is very intuitive. It's really designed in order to create reproducible and quantitative results. So you don't need to have a background in MR physics or experience with MRI to operate the system. The system does come preloaded with default sequences, but it also has the option to open up those sequences to make adjustments to parameters in order to customize the sequences to the needs of your study. And so you can really have a full flexibility with the system in terms of if you have someone who's strictly a biologist looking to acquire, biological data versus someone who has an interest in MR physics, have that flexibility with the sequences and the system to do that. And the system also allows you to optimize the protocols, save the protocols, and assemble protocol lists. So if you have a group or a specific study within your lab that you are running, 3 or 4 of the same sequences for each time point within that study, you're able to save those as a specific list and then load those in each time that you place an animal inside the magnet so that you end up with this reproducible imaging playlist that then you can for sure say that you've acquired with the exact same parameters to give you really that rigor and reproducibility that we're looking for with scientific studies. So just to kind of wrap up, the M-series systems really are designed with a preclinical researcher in mind, so you don't need to have an MR tech. Your average researcher can use the system. It does provide a streamlined workflow for animal preparation and positioning, and a user-friendly software interface. So it's very easy and quick to train on, and most people are able to acquire images by the end of the day. And we do also, with this system, enable a wide variety of applications, which we'll review. So before we get into the applications, we'll just do a quick audience poll.
Thanks, Dr. Wendell, for the fascinating presentation thus far. We would like to take a quick break here and ask you in the audience to tell us a little bit about the research you were doing. What is the main focus of your research? Is it cancer, neuroscience, cardiovascular biology, therapeutic research, nerve degeneration or modification, or MRI, or other? We would also like to ask, would you consider implementing MRI into your research? Are you currently using MRI in your research? Are you hoping to learn how to implement MRI into your research? Are you unsure how MRI would fit into your current research projects? Are you just here to learn? And is MRI not relevant to your research? So we'll take a couple more seconds for you to answer our poll questions. Great. Thank you all for taking some time to tell us a bit about more of yourselves. And now I'd like to return things to Dr. Wendell.
Thank you, Ivy. So next we'll talk about the key research applications of the M-series, which include anatomy and morphology, neurology, cancer biology, also cardiovascular biology. Contrast agent work is also a very popular application, as well as multimodal imaging and ex vivo imaging. We'll wrap it up. So firstly, we'll talk a little bit about anatomy and morphology. So this is just an example of the mouse abdomen. So this is the sort of image quality that we would typically expect. And these images are acquired in about 5 to 6 minutes. So we can see our T1 is located on the left, our T2 is located on the right. And so we can visualize the same basic anatomy with both sequences, but we do see some differences. So within the medulla of the kidney, we can see in our T1 that we have a darker area versus in our T2, a lighter area. So this difference in contrast is related to how the sequences are written. And this also can inform us a little bit about underlying tissue characteristics. So in certain situations where we may visualize like a tumor, we may also then visualize with a T1, you can see the anatomy, but with T2, we're able to see if there's potentially a necrotic core of that center to understand where fluid is. So these are one way that utilizing multiple sequences can tell us a little bit more information about the underlying tissue characteristics within the disease pathology that we're studying. These are some examples of spinal cord imaging. So the image on the left shows a fixed mouse that was acquired actually at the UIC training. And so we're able to visualize both the brain and spinal cord. And then some examples from live mice that are acquired with T2 images. So we're able to visualize both the brain and the spinal cord, and then the pathology in the rest of the body as well. So oftentimes with spinal cord imaging, we would take a sagittal plane, but oftentimes with abdominal imaging, we would do coronal or axial imaging as well. Some additional examples of anatomy and morphology include looking at hind limb inflammation. So in this case, this model induces acute inflammation by a topical application of an irritant. So we're able to segment out the inflammation, which is shown in red, and then we're able to also quantify the volume of the leg muscle, both in red and blue and green. And so then we can compare that using the animal as its own control. We can also segment out areas like fat. So in this example, we've done an automatic segmentation based on grayscale intensities of the visceral fat. We're then able to quantify that volume and then able to either produce this in a longitudinal study or look in response to treatment or exercise. So now to move on to some neuroimaging examples. There are some examples of the brain. So these are acquired in about 12 minutes. So this is just a standard wild type mouse where we're able to visualize the brain in 3 different planes. And depending on what the pathology or the area of the brain that you're looking at, utilizing one or more planes for image acquisition can be very helpful. Most of the time with neuroimaging, we acquire in a coronal plane, but oftentimes if you're looking for tumors, acquiring an axial or sagittal plane can also help you visualize the location of those tumors and any metastases that may have also grown in the meantime. So this is an example of a glioblastoma that we acquired through the UIC training that was performed a few weeks ago. So with this glioblastoma, we discussed that it was comorbid with oftentimes frequent edema. And so we acquired a T2 weighted image in about 6 minutes. And with the unknown, if it was how much fluid or if we had alterations of the ventricles, we ran a fluid attenuation protocol, which essentially is short for FLAIR. So we use that same T2 sequence. We're able to suppress the fluid. And then we were able to see these dark areas shown in the center image. And then we also can identify that as edema shown in red. And then when we apply that to a T2 weighted image, one of the slices that we acquired, you can see that the edema is shown there in red. The remaining abnormality in the brain then is identified as tumor. So this is one way that you can use multiple sequences to really understand the underlying tissue pathology within your models. So this is another example of glioblastoma. This is a longitudinal study. 4 days post-injection, we don't see any tumor growth at that point. All the structures appear to be normal. 15 days post-injection, though, we do see the growth of a tumor within the ventricles, and we are also able to visualize some metastases that have progressed throughout the ventricles. And we're able to then quantify that tumor volume in a third-party software, which we typically use VivoQuant. So another example from neuroimaging are studies in traumatic brain injury. So this particular example is using a percussion injury to the skull. So we're able to visualize that inflammatory lesion, particularly very early on after traumatic brain injury or stroke. We have accumulation of edema. We're able to visualize that over time, really to understand the progression of your model or also the therapeutic effects introduced by the researcher. And so we're able to visualize this edema. Additionally, another example would be epilepsy, where after epileptic induction, 3 hours post, we don't see any changes really to the brain. But at 48 hours post-epileptic induction, we see this really bright area along the cortex, which is indicative of large amounts of edema. So again, this is just one way that we can monitor either progression or in response to treatments non-invasively and also over time. So another common application within neuroimaging is stroke. So there are many different types of stroke models. This particular case is photothrombosis. So we can see the lesion quite brightly in these T2 images shown on the cortex. And we're able to quantify that over time. In this instance, this is a photothrombotic stroke. And then we're able to look at both T1 and T2 images. So the T1 is shown on the bottom, your T2 is shown on the top. So that bright area shows the lesion and is oftentimes indicative of inflammation. On the T1, on the lower panel, we're able to see those dark areas, which can help differentiate areas like edema versus hemorrhage versus dead tissue. So again, utilizing multiple sequences can really help differentiate and understand those underlying tissue characteristics. Next, we'll progress to cancer biology. So tumor detection is probably one of our most popular applications for this system, particularly because the system can be placed in the vivarium or in an existing imaging facility. You can really create high throughput with this system. So this example comes from the Institute of Cancer Research in London, UK, in which case they have many, many models of different types of cancer. And so they're screening multiple animals throughout the day, really looking for high throughput and not long imaging scans. So their scans run anywhere from 2 minutes to 12 minutes, depending on the resolution, the area of the body that they're looking at, in order to really increase the throughput and understand the progression against different drugs. So with the tumor response, oftentimes we see that the system's used for rapid screening. In the case of the ICR in London, their mice, are pretty much back in the cage within 5 minutes. So it's quite a rapid screening for them. With either deep-seated tissues or tumors, it can reduce the bias compared to physical palpation. If you have an abnormally shaped tumor, this again can help kind of reduce the amount of bias that you may have with physical palpation. The person who typically does your palpations are not in that day. This can help reduce that variability. Also can reduce the amount of animals used. So you can use the animal as its own control or look at disease progression within that same animal, reduce the amount of animals that you would typically use for either histology or other methods in order to have a really enhanced data curation. So this is an example of a flank tumor that we acquired at the UIC training a few weeks ago. So we acquired both with T1 and T2 weighted imaging. We're able to visualize the anatomy with both T1 and T2, but we are also able to see what appears to be a blood vessel in through the T1 infiltrating through that tumor. A similar model, the xenograft tumor model, we're able to essentially implant a tumor into the hind limb, identify those borders with either T1 or T2, and then quantify that over time. So in this case, the tumor volume, 730 millimeters cubed. This was quite a cool new application for me, nerve sheath tumors, which I've not really had the chance to work with before. And so from what I learned, essentially the Schwann cells continue to grow. And so they end up being enlarged over peripheral nerves. And so we can see here a pan through of the brain and the spinal cord, and then looking at specific slices. In the middle panel, we can see that bright area behind the lungs along the spinal cord shown in red. We could then quantify that, and then these two in the second panel as well. We're able to then pull that into analysis software to delineate the borders of those nerve sheath tumors or growths, and then quantify those volumes. So another example of a tumor model, it's a cervical tumor model in this case, where our controls exhibit these tumors and then we have a treated group. we can see that the tumor volume is smaller in the treated group compared to that of the controls. And we're able to take those animals out multiple weeks, assess the effectiveness of these treatments on tumor volume. Additionally, it's very complementary with other imaging other imaging methodologies like bioluminescence. So with bioluminescence, you can detect the presence of tumors or metastases. So you're able to localize the location of the tumors. With MRI, you're then able to quantify the volume, the progression of this. And so they're oftentimes used in conjunction. Also, MRI can be used as a standalone technique where you have tumor models where luciferase tagging is not possible. So lots of different options in terms of models that are applicable to use with this system. So next we'll move into cardiovascular biology. So these are some example images of a long axis view of the mouse heart. And so we can take both long axis and short axis views. People use this oftentimes either to replace echo or ultrasound, or as one way to reduce user variability. Oftentimes, people who are very experienced with echo find it very quick to do that, but new users often may struggle. So this is one way that helps kind of reduce the finesse that would be needed to typically acquire these types of data. So here's some example data from a short axis view. So we oftentimes pull this data into a third-party software called Segment, and then we're able to outline the myocardium for both the left and the right ventricles, and then track it over time throughout the cardiac cycle. Then we're able to export pretty much all this data, which includes diastolic volume, systolic volume, stroke volume, ejection fraction for both the left and right ventricles. With the long axis view, we can also get strain. So we're often able to, again, outline the myocardium in the long axis, and then we're able to get different strain metrics like mean strain, segmental strain, torsion, a wide variety of metrics that you would be able to analyze from a very quick MRI image acquisition. We can also acquire angiography. So this is a time of flight sequence that come on the system. So we're able to essentially map blood vessels in the body and the brain without the use of contrast agents. So here's a 3D reconstruction of the vasculature, some of the main vasculature in the brain, as well as the cardiovascular system. So next we'll progress into contrast agents. So contrast agents are a common application on this system, especially gadolinium-based contrast agents. Part of that is because gadolinium-based contrast agents have an enhancement based on the low field that the M series has of 1T. So you have quite a lot of enhancement compared to other systems. So this is an example of a 50 microliter injection of Primivos, which is a gadolinium-based contrast agent that was injected through the tail vein. So as we can see, as these images pan together, areas like kidney are much more enhanced. We can see that bright area from the gadolinium, and we can acquire these images as pre and post contrast. And utilizing a pre and post contrast acquisition, we're able to do subtraction in third-party software and get those areas of enhancement. So this is oftentimes used to identify areas with large vasculature like tumors or leaky blood-brain barrier, stuff like that. Also myocardial infarction, where you might have the leaking of gadolinium into other areas, either upon injection or upon clearing. We're able to analyze both pre and post and understand areas where we potentially have a compromised vascular system. We can also do dynamic imaging. So this is oftentimes performed while the animal's inside the magnet. So as I mentioned before, there's a small port where we can run a catheter to do dynamic imaging, injection of contrast or other agents while the animal's inside the magnet. So this particular sequence is acquired essentially 1 frame per second. So you're able to see the gadolinium as it flows in, in through the kidneys and in through the liver. So this oftentimes, again, is used for stuff like tumors, for cardiac imaging as well. So lots of options. Um, um, Aside from like these sort of applications, development of contrast agents is quite popular as well. So targeted contrast agent development is very popular, that you would do with well plates or not necessarily start in vivo animals, but essentially take it all the way from synthesis into in vivo. So next we'll move on to multimodal imaging. So the system does have the ability to be multimodal. So in our larger system, which is the M7, we have the ability to do simultaneous PET and MR. With our smaller systems like the M5 and the M3, we have the ability to do sequential imaging. So we have a PET insert that can be either placed on the side, where you can then acquire your MR with the exact same positioning, or the exact same animal bed. You can also then insert them into the PET insert in order to get the functional information that you would get from PET, which oftentimes provides you a little bit more information about, disease progression and metabolism in the body. So in this particular example, we're looking at hot rod phantoms. So this just shows that we can delineate down to 0.75 millimeters using our OSEM algorithm with the PET insert. And then this is a quick example of tumor hypoxia. So on the bottom, we can see our T2 weighted images show the visualization of a tumor in mouse. With the PET, we can see that there is difference in metabolism that we don't necessarily see with the anatomical change within MRI. So oftentimes this combination of information can help you understand functional changes before potentially anatomical changes that you would assess with MRI. So another example of using PET and MRI or CT. So with your T1, you're able to identify essentially the presence of the tumor. Utilizing PET, you can get that functional component. When you do an overlay, in which case we can visualize in the fourth panel that we actually have an area of missing PET signal, which would tell us that there's no functional or metabolic activity occurring there. Utilizing a T2 sequence then with that bright area would indicate to us that there's a necrotic core, which then explains why we don't have any metabolism occurring in that area if the tissue in the center is dead. So combining this imaging modalities really can help provide additional information and context about your models and what the disease progression or treatment looks like. And so oftentimes M-series is used with other imaging modalities, like the example we reviewed earlier of the bioluminescence. It's also common with ultrasound for cardiac as well as tumor volumes. So we can see it's very complementary with other imaging modalities that you may currently use in your lab. And this is also one way to just confirm and further understand your models and the underlying pathology. Next, we'll move on to ex vivo imaging. So ex vivo imaging essentially is taking excised tissue. So after the animal's been sacrificed, you're able to harvest that tissue, you can fix it, and then image it at a later time. So the advantage with ex vivo imaging is that you don't necessarily have to worry about animal welfare. So you don't have to monitor their vital signs, you don't have to deliver anesthesia. There's more ability to scan for longer periods of time. And so you're able to then acquire really high resolution scans with longer scan times with ex vivo tissue. And then you can really get nice images of very detailed structures. So areas like the brain is very common to do ex vivo, particularly after a longitudinal study. If you have multiple time points where you're concerned about either animal health and you'd like to have a quick scan after the animal's been sacrificed, you then can take that tissue, image for longer periods of time, and then go back and co-register. So that's one common way that ex vivo imaging can be integrated even within a longitudinal study. Additionally, ex vivo imaging is used to help guide histology. So oftentimes histology can be arduous for people. So especially sectioning a large tissue, looking for specific lesions. You have people that oftentimes will scan that tissue in the MRI, identify where those lesions will be located, and use that to guide their sectioning and staining. So this just helps streamline the workflow within your lab to spend less time potentially on your histology and understanding the underlying tissue characteristics. So with that, we reviewed a little bit of applications in all these areas: anatomy and morphology, cancer, cardiovascular, even multimodal imaging, and ex vivo options that you can use with the M series. So hopefully I've displayed that the M series really are designed for any researcher in the preclinical space. You don't need to have a tech to operate this system. The streamlined workflow and animal setup really does help, your average researcher without any experience to easily integrate this modality into their lab. And the software is very user-friendly, very quick training. And there is quite a wide variety of applications that can contribute to your research. So hopefully you will integrate this into your research, into your workflow, see how this can contribute and accelerate your work. And with that, I'd like to thank you all for your attention. And now we'll take any questions.
Thank you, Dr. Wendell, for the insightful presentation. Before we start our Q&A, we'd like to open the floor to Mark Sanders and Steve Schnell for any remarks.
Thanks. Mark Sanders here. So my video is blank, but that's just fine. The important things have been seen. So I'm very excited to be part of this presentation and be part of the effort that was started nearly 2 years ago to have this system acquired and placed in the UIC. And we're very excited here to have it hit go. Steve Schnell is the small animal lead that will be working with individuals to do the teaching and training. And if Steve is available, perhaps he could mention how you can get started in that process.
Yes, absolutely. Thank you, Mark. And thank you, Kara, for a wonderful presentation. For anyone who's interested in learning more about the system, I encourage you to visit our website at uic.umn.edu. There you can enter your training requests, and then we can get back to you and tailor the session more to your situation. So yeah, that's the first place you're going to go. Is you're going to request a training request, and then I will get back to you and we will discuss anything to do with your particular experiments. The BSL safety requirements we are BSL2 certified laboratory, so you may bring animals there. And yeah, I think that's the first place you're going to go, to our website. Anything else, Mark?
No, thanks. Again, we have boilerplate documents that we can help you with your IACUC protocols to make sure that that is as seamless as possible. And of course, we have the fantastic technical support by the hosts of our webinar today that we can fall back on, in addition to a number of the other correlative imaging modalities that were mentioned throughout this presentation. And that includes the IVUS spectrum for bioluminescence and fluorescence, the Vivo 2100 and F2, and our MicroPET micro CT that's located at our CCRB facilities, all utilize the same, if not identical, softwares to do correlative data here. So we're super excited about the compatibility in this port. Without further ado, I just really want to acknowledge and give special thanks to Dr. David Largaspada and the Department of Pediatrics and the Masonic Cancer Center for their support and contributions to get this system in place, not only for their fantastic work and efforts, but to share it more broadly to the university. So thank you very much to that group. And I'd love to hear any questions either on how to get in to use it or specifics for the M5 for any of us here on the panel.
Great, thank you, Mark and Steve. So now we'll take some time to go through the questions. Remember, the Q&A section is still open, so please send in your questions. Dr. Wendell, we have a question for you. Is the body coil M3 only?
So there are body coils for each of the M series. So there are for M3, M5, and then the M7. So the M3 and the M7, the body coils work slightly differently where they slide over the animal bed. But in the instance for the M5, the body coils are inserted through the back flange, and then the animal handling system is inserted through the front. So those are both still automatically tuned and matched. So that component of the ease of use is still applicable across all of the magnets.
Thank you. We have another question. How long does it typically take to acquire the images?
So the length of image acquisition really depends on what the goal of the data is. So in the instance you're looking to do tumor screening or detection, it could range, for only 2 minutes. If you're looking for very specific anatomy or a very small region in the brain, it can range based on the resolution you're looking for. So really can range anywhere from 2 minutes to 15 minutes, depending, again, on the application that you're looking for. Myself and Steve also can help everyone kind of go through that and understand what the parameters mean and the sort of resolution that you would hopefully need to delineate the particular area of the body or anatomy to visualize that with these sequences.
Thank you. And what are the performance stats of the system, TE, TR, and the gradient field range?
So the TE, TR, and the gradient field range. So the TR and the TE, you can program in with your sequences. So all these sequences are open on the front end when you acquire. So you're able to make those adjustments, those changes, particularly if you have either an interest or a background in MR physics or sequence composition and writing. All that stuff is accessible either through the quick access tab or even through the advanced parameters. You can make those adjustments. And then there's also preloaded sequences that come on that. In terms of the gradient field range, so the system operates at 1T. I also have a spec sheet that I can share with Steve if you'd like to share that with everyone. That goes through like the gradient strength, the slew rate, all of that information as well.
And what is the downtime processing multiple mice? So how long does it take to switch from one mouse to the next?
It doesn't, well, I guess it depends. The more you do it, the easier it gets, I will say. For me, it's pretty quick. I prefer to anesthetize the next animal while the first animal is essentially being imaged. That really helps increase the amount or decrease the amount of time that it takes to pull the animal out and make those adjustments. But it's fairly quick. If the animal's anesthetized, you can pull out the animal handling system, remove the mouse, put the next mouse in, apply the eye lube, and then place in the magnet. So it can be very quick, especially if you're imaging either the same area of the body, the same organ. It's very quick. Oh, Ivy, you're muted.
Speak on the topic of imaging. Does fur shaving improve the image somewhat?
No, so there's no interference from the fur. Essentially, because the way that MR works has to be a proton-filled area. So fur doesn't necessarily impact the image whatsoever. Areas like the lungs or essentially air-filled organs don't have much protons, so you don't have as much signal. So that's more the interaction, or the effect is mostly if you have a lack of protons or if the tissue's been dehydrated through after ex vivo. Some people dehydrate tissue for long-term storage. So that pulls the water out of the organs. That you'll have more effect than something like fur.
Thank you. This is actually a question Steve or Mark could answer. Where is the system physically at UIC, and can this be moved between buildings?
Sorry, could you say that again, please?
Where is the system physically at UIC, and can this be moved between buildings?
It could be moved. It won't be. It is in the first floor of Jackson Hall on the north side. We have a dedicated room just for this facility and its use.
Thank you. And another question for Dr. Wendell. How long does the tumor volume calculation take on average? And how large are the image files? Where will the UMN users store or access their data?
Okay, so I'll tackle the first portion of that in terms of tumor volume calculation. How long does it take? It really depends. So within VivoQuant, which they have access to at the UIC, there's various tools where it can help you outline or threshold to help streamline the essentially analysis. In my opinion, it can go fairly quick. It just depends on, again, the tools that you want to use to do your analysis. I would typically say like whatever you choose to do it across all the time points, across all of the participants or animals within your study. What was the other sections of the question? Sorry. How large are the image files?
Yes.
So the image files are pretty small, surprisingly. I think, Steve, we also discussed this when we looked at how large the exported data is. So they're fairly small. They're easy to transport. I have just your standard like USB key. I have hundreds of scans on it. So because the data is very small and compressed. And then, Steve, do you want to speak on where UMN users will store or access their data?
Yeah, absolutely. We have a transfer server that's available for all of our UIC workers or users. You just can put it on the transfer server, and then you pull it off and put it in your lab's data management program.
Great. And back to Dr. Wendell, is the data analysis tied to the instrument, or can it be executed on separate machines?
So the data analysis essentially can be done on any computer in theory. So all of the images are exported as DICOM. They also have the option to export as like JPEG and TIFF, other image file formats. So most of the time we would recommend exporting them as DICOM, but you can pull them into either VivoQuant, which the UIC has, or into other third-party softwares, even into something like ImageJ, in order to do data analysis. So it really is up to you in terms of once you acquire images, how you choose to process them. The UIC will have VivoQuant available, but there are also other options.
And back to Steve. Is the UIC planning to offer a storage space, or is it up to the user to retain all the data?
All the data that is generated by any of our instruments is the responsibility of the user. We do provide, as I mentioned, a method of transferring them from the instrument to a transfer server, and then each user can then pull them off from there and again put them in their own lab management and data management program. Very easy to do. Very easy.
Thank you, Steve. And last question for Dr. Wendell. I know you mentioned bioluminescence imaging. Is there fluorescence imaging capability as well?
So for the MRI, there's no bioluminescence or fluorescence that can be acquired at the same time. However, if you use contrast agents, oftentimes it can be used similarly to like essentially a fluorophore. And so with the use of gadolinium or iron oxide nanoparticles, oftentimes if they're targeted, we can hone them to specific areas of the brain and see those differentiation either in those organs or where they accumulate. So there's a couple of examples from Texas Children's who have done contrast agent work that is targeted to essentially tau protein in the brain. So they were able to look at tau load in Alzheimer's animals. So you can use it similarly to how you would, but you wouldn't get the histology level resolution that you would get using fluorescence, and bioluminescence can't necessarily be acquired at the same time, but more sequentially.
And just to build on that, the University Imaging Centers at the Jackson Hall location and our Cancer and Cardiovascular Research Building location have an IVUS spectrum that has the ability to do both bioluminescence and fluorescence imaging of small animals. And the tools and experience to help correlate that data will be available.
Thank you, Mark. We've reached the end of our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any outstanding questions in the written script, and we'll work to get this out to you over the next few days. I'd like to thank Dr. Wendell for this presentation today, as well as for taking her time, as well as for taking the time to address so many of your questions. Thank you, Steve and Mark, as well. I trust that we've all been able to provide you with some relevant information to the studies you're conducting back at your own labs. In the few days and weeks to come, if you have any further questions about the work that we discussed today, or questions about any of our equipment, including the M-series, I encourage you to reach out to us here at Syntica, and we will be happy to discuss this further. We would welcome the opportunity to discuss your specific research goals and how much of our equipment could help you move your work forward. Thanks again to all of you for taking the time out of your day to attend our session. We look forward to seeing you at future Syntica events. Have a great day.
Thank you, Ivy.
Thank you all.